Statistical analysis in oncology trials explained simply with the help of real case study. Can statistics save lives? Real oncology case example. Can this new cancer drug save lives? Introduction. Imagine a pharmaceutical company developing a new cancer treatment that could potentially help patients live longer than existing therapies. Before doctors can prescribe it, researchers must prove scientifically that the treatment works and is safe. This is where statistical analysis becomes critical. In oncology clinical trials, statistics transform thousands of patient records into meaningful evidence that regulators, physicians, and patients can trust. Here's a real world style oncology case study, and let's see how statistical analysis helps determine whether a cancer treatment truly makes a difference. 600 patients, 24 months. One answer. Here's a real world example with a study objective. Say a pharmaceutical company is testing a new drug called ONCO101 for patients with advanced lung cancer. So the study design here is phase 3 randomized clinical trial. 600 patients are enrolled. 300 patients they receive 101 treatment, and the another 300 patients they receive standard therapy, and the follow up period is 24 months. Who gets the new drug? Now here the primary endpoint is overall survival, which is OS. So the question here is, do patients receiving 101 live longer than patients receiving standard therapy? Now here the secondary endpoints are PFS, which is progression free survival, objective response rate ORR, safety profile, and quality of life. I have also made one other video looking into the difference between PFS and OS. You can always go through and find out that specific video, the difference between PFS and OS in oncology. Millions of data points are collected. Now the step 1 is data collection. Now researchers collect data from every patient. After 2 years or specific number where the clinical trial study duration is executed, the database contains millions of data points. Statistics helps convert this raw data into evidence. It's not mandatory that every oncology clinical trial will have 2 years. This is just an example case study which I am referring to, right? It could be even 6 months, or it could be 5 years, or it could be even 10 years. So this is just an example real case study. So the variable here is data captured is age, gender, cancer stage, which is stage 1, 2, 3, 4, the treatment group, whether it's a standard therapy or onco 101, the date of randomization, the date of progression, and the date of death, and what were the adverse events. And this is just an example. The data collected is not limited to this specific window. There are many data set collected, but this is just a few examples as to what primary inputs that are collected. Who lived longer, the patients in standard therapy or in new therapy? That is what the investigation is in this is real case study. Now step 2 is defining analysis populations. Before analysis begins, statisticians define who will be included, whether it could be ITT, MITT, safety population, or PP population. So the definition of this analysis population, it should be clear in the statistical analysis plan when it is drafted, and it should be finalized before approval or before signature of that statistical analysis plan. Now ITT includes all randomized patients. So here there are two groups, Onco 101 and standard therapy. Patients are distributed 300 in Onco 101 and standard therapy 300. Regulatory agencies typically rely on ITT analysis because they reflect real world practice. Now in the safety population, those patients are included who received at least one dose of study treatment, and this population is generally used for the safety analysis. Are the groups really equal? Step 3 is baseline characteristics. Now, this is a very important point that statisticians they verify whether the two groups Onco 101 and standard therapy whether they are similar at baseline. So that is the first thing the statisticians do before comparing the treatments. Now, why this is done? It is done because you should ensure that there is a balance maintained between these two groups. Now, Onco 101 is 300 patients and standard therapy is 300 patients. So, these two groups they should have a balance so that a fair comparison is done and there is no biases included. Now, if one group had significantly healthier patients, survival differences might be misleading. So, say one group is having healthy and the another group is not healthy, so what happens in that scenario? The survival differences you will see. One patient will always, all patients they will always be dying early. There is no survival chances. There is no hope. So to ensure that the fair comparison is done, there is no biases. This is a very important step for biostatisticians to ensure that these two groups are similar at baseline. Now I will explain this. Mean age is 61.8 and standard therapy is 62.1. So there is plus or minus 1 or 2 differences, which is fine. That means these two mean age of these two specific groups, it's somewhat similar but not that too far. Plus or minus 1 or 2 is okay. So male percentage is 58 and standard therapy it is 57, plus or minus 1%, which is okay. Stage 4. So in Onco 101, there are 82 patients which are at stage 4, whereas in standard therapy, there are 81 patients which are having stage 4, which is somewhat similar to extent, plus or minus 1. So this specific step, baseline characteristics, comparing treatments before, is very important that these two groups should be similar at baseline. Then only you can do the fair comparison and you can get the correct results. Plus 5.3 months longer life. Now step 4 is overall survival analysis. Now this is very important analysis and the results is 24 months and these two groups, Onco 101 and standard therapy, after 24 months, they are having the results of median overall survival of 20.5 months and 15.2 months, respectively, right? So the difference between Onco 101 and standard therapy is 20.5 minus 15.2, which is 5.3 months longer than on average. That means patients who had this Onco 101 treatment, they are approximately having 5 months more survival than as compared to the standard therapy, right? So those patients who did consume the treatment Onco 101, their survival chances of living in this world, it has increased to 5.3 months, right? But statisticians must determine whether this difference is real or due to chance. So you have got the data, you analyzed it, you got 5.3 months longer also with respect to the new treatment. But whether this difference is with real or due to chance, that is the next step. The graph that changed everything. We are talking about here is Kaplan-Meier survival curve, which is usually used in almost all the oncology clinical trials. Now this is a step 5. Now Kaplan-Meier curve displays patient survival over time. So here you will see survival percentages, and in the y-axis you will see survival probability, and the x-axis you will see the time. So conceptually, if the curve is higher, it's a better survival. If the curve is lower, then it shows poor survival. Separation between curves suggests treatment benefit. Now if the oncology 101 curve consistently stays above the standard therapy curve, it indicates improved survival. Now what happens here is, say this is the survival probability and this is the time, right? So this is time and this is the survival probability. Now if the onco curve is like, higher than the standard therapy, and say standard therapy is like this, right? Now you see it is staying consistently above to certain extent, to a certain time period, then it shows improved survival, right? Now what happens here is, say I will show another example here. Say this is the onco 110 curve, and the standard therapy is crossing. Now if you see this crossing, that means that is a mixed results, right? The onco 101 treatment in this specific real case study is not showing improved survival, so it is not better treatment as compared to the standard therapy. So this is how you can correlate. So curve, survival curve, it gives a very meaningful analysis, and especially Kaplan-Meier is the powerful tool in the oncology trials that you can use, and without this curve you cannot do any US FDA submission and any regulatory submission for any clinical trial. Kaplan-Meier curve has to be there, and it's a mandatory and a compulsory curve, like that you will see in almost all oncology clinical trials, be it overall ovarian cancer or prostate cancer or any type of cancer clinical trial study. P is equal to 0.03. What does this indicate? Does it indicate highly significant, or does it indicate highly insignificant? Now step 6 is log rank test. This is also one more powerful tool which is generally used in oncology clinical trials. Now the log rank test compares the survival curves statistically, right? So the results is p value 0.03, which is less than 0.05. That is the alpha, that is the level of significance, right? Hence the difference is statistically significant. So this means that the survival improvement is unlikely to be due to random chance. So what happens is you collected the data, then you did this Kaplan-Meier curve. Now once that curve is much higher than the standard therapy curve, then you need to test again whether it is due to chance or it is a real effect, right? So the next step is to do the p-value test, that is the log rank test. Now once you do this test, you have statistically proved that there is a significant difference, right? 28% lower risk of death. Step 7 is hazard ratio HR. This is also one of the important tool to be used in oncology clinical trials. Now the result here is, for this specific example, is 0.72. The interpretation is patients receiving Onco 101 experience 28% lower risk of death as compared with standard therapy. Now the confidence interval here, 95% is 0.6 hyphen 0.86, because the entire confidence interval is below 1, that is this confidence interval is below 1, the treatment benefit is statistically convincing. Now what happens is, whenever you see this hazard ratio, it should always be between these two values, okay? So that is very important. Whenever you do the value, that is hazard ratio, it should be between the 95% confidence interval, then only you are getting the correct data, else it's a garbage kind of data that you're analyzing. So it's very important, and there are various techniques that you can choose. So if the hazard ratio is 0.72, you do 1 minus 0.72, right, which will give you 0.28, right? It will give you 0.28. So that 0.28 into 100 is your 28% lower risk of death. That is how it is calculated. Cancer slowed down. Now step 8 is progression free survival, which is PFS. Now researchers also evaluate how long patients remain free from cancer progression. Now the terminology of this specific secondary endpoint is PFS, progression free survival. Progression is cancer has progressed, means cancer has progressed means the tumor of that specific cancer has increased, right? When you add this free, that means the tumor specific in that cancer disease has not increased or it has not progressed. That is why it is called as progression free survival. That means the tumor in that specific cancer disease has not increased. It could be the same size or it could have been shrinkage, or it could be the less kind of centimeter or diameter, but the tumor has not increased. It is either same from the earlier or it has decreased. That is why it is called as progression free survival. Now this is a very common layman's statement language as to if anybody asks you this question in the interview, what does this progression free survival mean? So you should know how, from where this terminology is coming. So now see the results here is Onco 101 and the standard therapy here is 7.1 and the median PFS is 11.4 months. So there is the improvement of how much? 4.3. Now how does this 4.3 has come? 11.4 minus 7.1, which is equal to 4.3, right? Onco 1001 minus 7.1, 11.4 minus 7.1 is 4.3. Now this suggests Onco 101, it delays disease progression. Delays disease progression meaning it delays the tumor not to increase, either to stay in that specific size in the body or to shrinkage, right? The disease has not progressed. That means the tumor cells have not increased. They are into the same size or they are into the lesser size. Now these kind of terminologies is very important when you work in the oncology clinical trials. Now say for example, if the Onco 1001, if it is 6.1, right, that means Onco 1001 is not a better treatment. By consuming this Onco 1001 treatment, the disease has progressed, it is not better, right? So you need to play with numbers and you need to understand from this to the and making it understand for the common layman's language, or any person who would like to understand how does this 4.3 months came, why it is improvement, because it is plus sign. Now suppose if it is 6.1 and the standard therapy is 7.1, so when you subtract this new treatment minus standard therapy, you will see -1. So -1 is not improvement, it is degradation. It has not improved, right? So it has deteriorated. So this is how the meaning comes with the plus and minus signs. 50% response rate before treatment and after treatment. The tumor size was 8.2 centimeter before treatment, and after taking the treatment, the tumor size has decreased to 4.1. That means reduction of 50%, right? 8.2 minus 4.1 is giving you 50% reduction. That is how it is coming as 50% response rate. Now whenever you do any kind of oncology trials, the primary diameter is nothing but calculating the size of the tumor. That is very important. Based on the tumor size only you can play around with the oncology clinical trials. Now the step 9 here is objective response rate, ORR. Now researchers evaluate tumor shrinkage. Tumor shrinkage is nothing but whenever you are considering or taking the Onco 1001 treatment, that new treatment, Onco 1001 treatment, the tumor should decrease, it should shrinkage, their diameter should be less, right, as compared to the standard therapy. Now the results here, it shows complete response 8% as compared to 3%, partial response is 42% and 25%, total ORR is 50%, whereas the standard therapy is 28%. Now the interpretation here is half of the patients experienced meaningful tumor reduction with Onco 1001. Now whenever this specific ORR is calculated based on the complete response and the partial response, right? So the total ORR is 50%, 42 plus 8, which is 50%. That means half of the patients, the tumor has tumor has reduced in size after consuming the Onco 1001 treatment. I hope I am making myself very clear on this, because please understand, oncology is very interesting, and if you know how to play with these numbers and if you know how to interpret these numbers, life is very easy to work on oncology clinical trials. Remember one thing, but your fundamentals and the basic foundation should be clear, right? Is the drug safe? What are the potential risks and the safety results? Step 10 is safety analysis. Now, effective alone is not the only factor for getting the approval for any oncology clinical trial from regulatory agencies, right? Researchers should also evaluate the safety. That also is an important factor to get your oncology clinical trial approved. Now the next thing is common adverse events and the serious adverse events. Now these are the only two important factors through which you can measure the safety analysis, right? So now the common adverse events are fatigue, nausea, anemia in Onco 1001 and standard therapy. Now when you compare these two treatment groups, 35, 28, 22, 18, 15, 14, plus or minus 1 or plus or minus 5, that's not much kind of difference. But say for example, this common adverse events anemia in Onco 1001 is 15, whereas in standard therapy it is 30 or 50, then it becomes the question. So there is not comparable or manageable kind of the data that you're getting, right? So the serious adverse events also, when you have these two treatment groups, Onco 101 and standard therapy, 8% are having serious adverse event, whereas as compared to standard therapy, it is having 7 percentage of patients who are having serious adverse events. So which is somewhat similar, not equal but somewhat similar, plus or minus 1 or plus or minus 5. That's not much kind of major difference like 35%, or here it is having 100%. So that is not the case here, but it is somewhat, the numbers are somewhat nearer. That is why the safety profile appears manageable and comparable. So that is how you need to analyze these numbers. Does it work for everyone? Everyone meaning across all groups and across all subgroups. Step 11 is subgroup analysis. Now researchers should also test whether the treatment benefits they remain consistent across subgroups, meaning male and female, when you compare the two subgroups, the hazard ratios should be somewhat equal, zero point 0.7, 0.75, which is the 0.01 difference, right? So it is 0.05 difference, male versus female. That means the treatment benefits are consistent between male and female. Whether it is consistent between age less than 65 and greater than 65. So those patients who are having less than 65, the hazard ratio is having 0.69, whereas in greater than 65 it is 0.74. That is why the consistent findings strengthen the confidence in the treatment effect. So that is why the subgroup analysis is done. Now you will feel, yeah, so many tables are I have to generate the total age, then age group less than 65, then greater than 65. So there are so many subgroup analysis, and then the programmers, they think, why there are so, what is the meaning of having subgroup? This is the meaning, because the treatment benefit should remain consistent. So any kind of your project manager or any biostatistician or any senior person is telling you that, okay, we are not getting the results, based on the gender overall total. Now your manager is telling you, can you also do the subgroup analysis? Now you will be keep thinking as to why this person is telling me. Now, if any person is telling you, or any sponsor or any kind of CRO person is telling you, there is some reason behind it. So if this is what is the reason: male versus female, smokers versus non-smokers, and different geographical regions. So the subgroups is unlimited. You can add as many subgroups if you want based on the available data that you have. You can also include the efficacy primary value, or any lab specific value, whether you want to see is the benefit of treatment is there between that specific lab value or between that specific efficacy value. So it is very important subgroup analysis. It breaks down further and it shows you the hazard rows for the two different categories which are there in the subgroup. Approved or rejected. Step 12 is conclusion. The primary endpoint is specifically the overall survival, which has significantly improved, which I just recently explained in the above slides. The secondary endpoints, which is PFS and the ORR, the progression free survival and the objective response rate respectively, are improved, which I clearly explained in the above slides. The safety parameter is an acceptable safety profile, which I already explained that there is no difference, plus or minus 1 or plus or minus 5. That's not a major kind of a difference. So it's an acceptable safety profile. So the efficacy and safety, it is falling into that condition. So the regulatory interpretation is the evidence supports that Onco 1001 provides clinically meaningful benefit for patients with advanced lung cancer. Now, this is just a real case study which I have tried my best to explain in a very common layman's language, which is again a practical kind of a situation which I have explained you this kind of a numbers. Now remember one thing, theory is one thing and practical is something, like unless and until you get an opportunity to work with live data in any company with any kind of phase 1, 2, 3, 4, whichever cancer studies that you are having, the you will get more weightage and experience, and there will be still be many doubts and questions after explaining this. But when you work or live on the projects in any company, your all your questions, all your doubts and all your thinking process, everything will be cleared, but in this specific video I have done my best to explain as to what are the statistical analysis parameters that you need to use, what are the specific measures you need to take decision, and how you need to check whether the treatment has improved or it has deteriorated. So I have done my best, and I hope this video will help you in some way or the other in understanding and working on the oncology clinical trials. Now there are certain key takeaways. The number one is statistical analysis converts clinical trial data into reliable evidence. That is the importance of statistical analysis. Number two is overall survival is often the gold standard endpoint in oncology. So after taking the new treatment, what are the chances of survival? Uh, Living more in this world. So that is what is the gold standard endpoint. That is what we need to check. Step 3 is Kaplan-Meier curves visualize survival differences. And now a picture or a graph, it can make a big difference when you visualize that the chances of the specific new treatment is more than the lesser. So graph will give you the very clear picture based on the data that you have collected. Step 4 is log rank test determines statistical significance. So the Kaplan-Meier curve will visualize the picture for the data that you have collected. Log rank test, it will statistically it will test and it will give you the evidence that it is statistically significant. Hazard ratios quantify treatment benefit as to how the treatment is benefited as compared to the standard therapy. That is what is the role of the hazard ratio. Step 6 is safety analysis is equally important as efficacy. Now remember one thing, in any clinical trial, be it oncology or non-oncology or any clinical trial, safety and efficacy goes hand in hand. But before efficacy, safety comes first. Safety is a priority for any clinical trial. That drug or the medicine should be safe enough to consume for any human being. That you check first and then you do the efficacy analysis. Number 7 is regulators rely heavily on statistical evidence when approving cancer therapies. Now you go to any regulator agencies, you go to America, you go to Europe, you go to India, or you go to Australia, you go to China, Russia. So each country will have its own regulatory guidelines. So that clinical trial is specific to which country. So based on that specific country, you need to follow the regulatory guidelines, and those regulatory guidelines which you have in that specific country specific, they usually rely on statistical evidence. So whenever you do your data, it is a very responsibility and accountability kind of a position that you're working as a SAS programmer or a biostatistician in any industry. Designation does not matter, even if you're working as a manager, senior manager, or a director, and if you fail in doing the statistical analysis and the regulators don't accept a clinical trial, your designation does not help you any longer. So the main purpose of working on this oncology clinical trials is how honestly, with integrity and the right data, you are kind of analyzing, right? So be it any SAS programmers and biostatisticians, they both have their own responsibility and accountability in their own role that they're working in any organization. So that comes with most kind of important factor when you work in any company. Now, the summary here is behind every approved cancer treatment lies years of clinical research and rigorous statistical analysis. Now, as you know, any treatment that comes into the market, it takes 5 years, 10 years, 20 years to get launched into the market, and it also consumes a huge amount of millions and trillions of money. And statistics is not just about numbers, it is a scientific language that helps researchers determine whether a treatment can truly improve and extend patients' lives. Now, in this specific video, I explained you Kaplan-Meier curves and hazard ratio, then the log rank test. So these are the tools of statistics that help you whether the treatment has improved or it has deteriorated, or whether the patient's lives are extending for their survival, or it is deteriorating, or they do not have time to survive in this world. So these are the various tools that will help you. So numbers are very interesting. If you love to play with numbers, then statistics is the subject for you to get master into it, right? For aspiring clinical programmers, biostatisticians, and clinical data professionals, understanding oncology statistics is one of the most valuable skills in modern drug development. Remember one thing: working in any organization as a SAS programmer, biostatistician, or clinical data professionals comes with responsibility and accountability, and these are the most valuable skills. And if you want to utilize in a better way, you need to make an effort in understanding as to what is an oncology. So that's how, the drug development happens in this oncology. So this is what is all about the statistical analysis in oncology clinical trials. The numbers that save lives. So we are playing with numbers, especially in statistics. We are using the tools, and based on the tools, we make a decision whether the treatment has benefited. And based on that submission to regulatory agencies, we are saving the lives and we are saving the patients, especially who are living in this world. So that is what is the responsibility and accountability that you carry when you work in this field. Many thanks for watching my video and happy learning. Please like, share, subscribe, leave a feedback comment. Many thanks for your support and having great connection.